Today is going to be on mechanisms of genetics. So we're going to be talking about a lot of stuff, including mutations, meiosis, and Punnett squares. A lot of you remember Punnett squares is not exactly your favorite. So you got to pay a lot of attention on this so that you're going over it and ready to do it on the test when you experience it. So our objective is that you will be able to understand, explain, and use the topics of mutation, genetic variation, and Punnett squares in the context of just normal explanation also in your test. So if you need to, you're going to go ahead and open up today's reference sheet, which is the cheat sheet that I've attached at the bottom. And our first section is going to be on mutations, which are random changes in the base sequence of DNA. And so again, this is the process of the central dogma, right? We start out with the DNA inside your nucleus. We do the process of transcription to turn it into an mRNA message so it can leave the nucleus. And then there's a ribosome that helps us with the process of translation that makes the code of mRNA into amino acid sequences. When connected, they're all a protein together. This process, sometimes there's mistakes. And when there's mistakes, we get mutations. It's like if you ever make mistakes while texting, right? When you text, you might type in the wrong key, or you might just be adding in extra letters when you don't mean to, or missing random letters where you're supposed to. And usually we can understand it, but sometimes it does make it difficult to understand the message, same way it is in transcription and translation. So when you type and you type the wrong key, these mistakes in DNA are called mutations. So for example, if you compare a normal gene to a mutated gene, you'll make a normal mRNA through transcription, and then you'll make a normal amino acid sequence through translation, and you'll get a normal protein at the end, which is the product you want. On the other hand, if you have a mutated gene, you can get mutated mRNA and mutated amino acids, and you can get an abnormal protein, or sometimes it can just result in no protein at all. There are different types of mutations. For example, if we're comparing this licorice and gummy bear setup of DNA, you can have a substitution where instead of where it used to be green and white gummy bears, we have red and yellow gummy bears, right? They've been replaced or substituted. We can have a deletion event where there's supposed to be a code here in the original sequence, but it's absent here. We can also have an addition event where down here, there's only supposed to be 2 rows of base pairs, but there's an extra one added in. Here you can kind of see as an example, if the original DNA was going to code for, an mRNA sequence that said UUA, that would correspond with the amino acid leucine. But if you got a frame shift mutation, like down here, every amino acid afterwards is going to be affected because you inserted a G here. So now it reads UUG, and the next triplet codon is going to start with an A instead of whatever it was originally going to start with, even though this A should have originally been here with no G. You can have a silent mutation where you change one of them, like for example, you change the first U to a C in the mRNA, but CUA also produces leucine, so no biggie. You can also have a missense mutation, which swaps one of the bases for something that causes it to be the wrong amino acid. Still an amino acid, it's just the wrong one. And then you can have a nonsense mutation where you trade one of the letters and you turn the codon into a stop codon, and that prematurely terminates or cuts off the amino acid sequence being made. So the protein now either is not proper shape, doesn't have the proper length, or doesn't even exist because you cut it off too early and now it isn't a functional protein. So will the mutation lead to changes in the offspring? Well, if the mutation is in your body cell or your somatic cell, then no, it's just in you. But if the mutation happens in an egg cell or a sperm cell, then yeah, that will affect the next generation. Section 2 we're going to talk about is meiosis. And again, some of you think it's how babies are made, but it's not really how babies are made. It's how eggs are made, right? Eggs and sperm. As a human, all of our somatic body cells have 46 chromosomes. So our neurons, our nerve cells, our epithelial or skin cells, our red blood cells technically actually don't have DNA in them. White blood cells do, but red blood cells don't. Smooth muscle cells also do, right? They all have 46. These are our body cells. They all have 46 chromosomes as a normal human should. But sperm and egg must fuse together to create one human cell that is the first cell of a baby. So their number is going to be different, right? they have to have a certain number of chromosomes in the egg, a certain number of chromosomes in the sperm, so that when they add together, they make 46 for the baby. And that's 23 and 23. So how do we even get to those numbers from the 46? Well, meiosis has two divisions, which will create the gametes that have half as many chromosomes as somatic cells. So we'll go through meiosis 1, where you have the original number of cells, and you actually cut the number in half by splitting them into 2 cells. Now, those halved numbers will go on and go through meiosis 2, which is very similar to the normal process of mitosis, where you end up with 1 chromosome in each cell of 4, right? But originally, each cell had 2 in this case. For us, it would be 46 and 23. In each cell, originally there were 2, but in the gametes produced by meiosis, there's only 1 in each, which is exactly what you want. Meiosis will create genetically diverse offspring because of a process called crossing over, where when the homologous pairs are lined up together in meiosis 1 at the beginning in prophase and metaphase, they will actually swap chunks with each other and create brand new genetic combinations that have never existed in any time of history, which is really cool because it means that you are an original. Mendel's law of independent assortment also helps out with diversity and uniqueness, and it says that traits that are inherited independently will separate independently, right? So that means a couple where one parent has freckles and the other has curly hair can have a child with curly hair and freckles, or have a child that has no curly hair and no freckles, or a child that just has curly hair, or a child that just has freckles with straight hair. So these traits are all separate. They don't depend on each other to separate. So you can have any kind of combination of these traits on offspring. So they're independently assorted. Here's a diagram overall of just what's happening in meiosis. So you're starting out with a normal cell with a normal amount of chromosomes, some from the dad, some from the mom, and they double to prepare for meiosis to go through, right? This is happening in interphase. And then the homologous pairs will line up together and split apart. So when you originally have 4, you now only have 2 and 2. And then those will split the sister chromatids in half along their vertical bisector, and you'll only have 2 chromatids in each cell at the end of meiosis 2. Answer this question for me now in the pair deck. Each body cell of a goat contains 60 chromosomes. So how many chromosomes would there be inside of a goat gamete? The answer is that if a body cell is 60 chromosomes, the gametes should have half the number. So 60 divided by 2 should be 30 chromosomes in the gametes or the sex cells. So the answer is B, 30. Genetic diversity is known as the differences among any particular species, for example, cats. In a single litter of kittens with the same parents, no 2 kittens look exactly alike, and that's the diversity of genetics. Which of the following are features of meiosis that increase genetic variation? There are technically 2 answers. So the answer choices are both C and D. Crossing over and independent assortment will increase genetic variation and make you unique to anybody else that exists in the world and any animal compared to any other animal inside their species, because these 2 things are creating new genetic combinations every single generation. Okay, third section is going to be Punnett squares. So basically we use these to produce probabilities of what are the odds of 2 people mating and producing children with specific combinations of specific traits. So as a vocab refresher, a trait is a characteristic. It's some kind of descriptive characteristic, for example, brown hair, being short, having wrinkles. Heredity is when genes are passed from parents to offspring. So when you inherit something. A gene is a specific chunk on a specific place of your DNA in your chromosome, and it has a specific purpose or job that it produces a protein for. So for example, the BRCA1 gene is in charge of preventing breast cancer. And if you have a mutation in your BRCA1 gene, then you will get breast cancer. An allele, for example, is all forms of the same type of gene. So some genes have 2 alleles, a dominant and a recessive. Some have multiple, like blood type, A, B, O, and some have many, many different alleles. For example, genes that contain or control your skin tone or your hair type. And These forms are all called alleles. So a gene is that specific area of a chromosome that specifically tells you what type of trait you're coding for, but the allele are just the different possible versions of that gene that you can have. Here is a karyotype, and in every karyotype, you should have 2 chromosomes of each type, right? They're homologous pairs. And one of those came from your dad, marked in green here, and one of those came from your mom, marked in purple. And this is a human karyotype. It is a full normal set because there are 2 chromosomes per there are 2 chromosomes per type of chromosome. And then also this is a male individual because there's an X chromosome and a Y chromosome. If it were both X's, like 2 large ones, then it would be a girl. So based on the fact that you get 1 chromosome from your mom and your dad, each gene you have, 1 is from your mom and 1 is from your dad. So those are different combinations sometimes. So sometimes they're the same type of copies, but one of the alleles is from your mom, one of the alleles is from your dad. So sometimes an allele is dominant and the other one is recessive. Dominant means that it's stronger. So we represent it using a capital letter, and it's the one that's going to express or show itself. And a recessive is a weaker allele, so it's going to be represented with a lowercase, and it's going to not show if there is a dominant. It'll only show if there are 2 of them present. I'm going to ask you here to go ahead and drag a blue star over the genotypes that would show a recessive trait, like I explained. The answer should be only where both letters or both alleles are recessive. Then you will show a recessive phenotype. If there's even just 1 single capital allele or 1 dominant allele, then the dominant phenotype or dominant trait is going to show. So here's what I was referring to. The phenotype is the physical trait that is visibly seen and determined by the genotype. So the genotype and the environment are what determine what the phenotype looks like or the physical trait looks like. The genotype is specifically just the combination of the genes or alleles that determine the phenotype. So the genotype is your genes, the phenotype is your physical trait. Your genotype tells you what your physical trait is probably going to be. So identify each of these. Please tell me if these alleles are a genotype or a phenotype. Use the red check marks to tell me if it's a genotype and use the blue check mark to tell me if it's a phenotype. Okay, we have genotype for AA. Brown eyes are a phenotype. TT is a genotype and red hair is a phenotype, right? Physical characteristics versus genetic combinations. So homozygous, when we talk about allele combinations, homozygous means that they are identical alleles for a single trait. You can have homozygous dominant, capital, or you can have homozygous recessive, lowercase. We also have a heterozygous combination of genes or alleles, which mean we have different alleles for the same trait, which would be capital and lowercase. So there's only 1 case of heterozygous, so we don't have to say heterozygous dominant or heterozygous recessive. It's only heterozygous. Here's a visual representation. Now, if you have the same gene on both the homologous chromosomes, then that's homozygous. If you have different genes or different versions of the gene, so they're different alleles, then it's heterozygous. So here we have heterozygous alleles, and here we have homozygous alleles. Notice that this is in the same area on the chromosome on each homologous pair. So therefore, it's the same type of gene, it's the same gene that's going to be coded for. But if they have different versions of them, then they're going to be different alleles. If they're different alleles, then they're heterozygous. So use the circle and mark all of the homozygous genotypes. They can be homozygous dominant or homozygous recessive. The answer should be where you have capital H, lowercase x, and capital Z. This one, and this one are all heterozygous. So here's our overall vocab that we just went over. And this is a Punnett square. This is where we're going to be using our genotypes and alleles. Punnett squares are used to predict what 2 parents' potential offspring will look or act like, sometimes traits or behaviors. Parent 1's genotype will be over here. Parent 2's genotype will be over here. Notice that the letters that were originally together in a pair are now separated into their own boxes. And then you'll pull them vertically down, horizontally over, and you're going to get combinations that tell you the possible genotypes for their children. So let's do a hair color example. If the trait for the hair color is that brown is a dominant color over red, and we have the parents being heterozygous and heterozygous, both brown haired because they have a dominant, then what are the chances of their child having red hair? Well, we want to find that out. So let's see. Let's put the moms, put the dads, and then we're going to make each potential offspring's genotypes. I'm going to ask that you fill in the Punnett square here with the bottom row. So pull this B down here and B over, B down here, B over. Now we know the genotypes. So you should be able to predict the phenotypes because you know about the hair color trait, right? So we know that brown is dominant over red. That means capital B is going to be brown hair because capital B is dominant. Capital B, lowercase b is going to be brown hair because capital B is dominant. But lowercase b is homozygous recessive. So that's going to be red hair because there's no capital B or brown coloring to cover it. So tell me, what are their chances of having a child with red hair? What percentage? Right, 1 out of 4 are expected to have red hair, which means that they are going to be 25% likely. So every time a child is born between this couple, there's a 25% chance that the child will have red hair, which is kind of crazy because they're both brown hair. So they're gonna be like, oh my God, where did this baby come from? So let's solve this. Cystic fibrosis is a hereditary disease that affects the respiratory and digestive systems. Cystic fibrosis occurs when 2 recessive genes, little c, are present. A person with 1 allele for cystic fibrosis is called a carrier of the disease. So if the mother is a carrier of the disease and a father is homozygous dominant, what are the chances that the child will be a carrier of cystic fibrosis? Well, you're going to have to set up a Punnett square with the big C, and then a big C, little c, because the mom is a carrier. Find out the percent chances that the child is also a carrier like the mom, which means that the child has to be big C, little c, right? So we're going to set up the Punnett square now. And solve. There's also dihybrid crosses. The ones we've been doing so far are monohybrid crosses. And dihybrid crosses are when you have 2 traits at a time, right? And we're combining them into one table to make it easy to track what's going on. So for example, we can have a round and yellow pea and a green and wrinkled pea, and we can do a dihybrid cross. And it's a simple one just because, well, they're both dominant or both recessive, depending on the trait. However, if you notice, instead of having 1 letter in each box, they now have 2 because in a dihybrid cross, you're talking about 2 different genes. So there's 2 different letters. So here's a basic problem. What are the possible genetic combinations among an offspring of a man who is heterozygous for both dimples and freckles and a woman who is recessive for dimples and freckles? Well, what is the first step that we have to do? We have to figure out what the parental genotypes are. So the dad is going to be heterozygous for both. So he's going to have capital and lowercase for freckles and capital and lowercase for dimples. But the mom we know is recessive for dimples. And we know she's also recessive for freckles. Okay, moving on. So now we're going to do the FOIL method. The FOIL method stands for first, outside, inside, last, and it's also a math technique. When you're doing FOIL, you're going to be pulling the first of each pair of letters. You do have to consider the letters separately, right? So you're first going to pull the first H and the first S for F. For the O, you're going to be pulling the outside H and the outside S. For the inside, you're going to be pulling the 2 that are on the inside, so the inside H and the inside S. And for last, you're going to be pulling the last H and the last S. So as you can see, we're pulling the first, outside, inside, and last, and that's the FOIL method. And you should have 4 gametes for each parent during your cross. So we're going to do a 4x4 cross. 4 times 4 is 16. So we're going to be working with 16 boxes now, right? So if you pull the dads, you have the first F and the first D, so capital, capital. outside F and outside D, capital F, lowercase d, inside F, inside D, in lowercase f, capital D, and then last F and last D, so lowercase f, lowercase d. Mom's is pretty easy though, because all four are the same, because she doesn't have any differences. They're all lowercase. First F, first D, outside F, outside D, inside F, inside D, last F, last D. So these are all the possible combinations of the alleles in the dad's and the mom's gametes. So her eggs can each contain this, and his sperm can each contain this. So let's set up our cross now. So we're putting the mom's 4 separately across the top and the dad's 4 separately across the bottom. Now we're going to pull them in. So if you pull these boxes together, yeah, I get the mom's on top, but there's a capital here. So the capitals always go first. So we have capital F, lowercase f, capital D, lowercase d. We always have the capitals go first. So then you would finish it out by pulling them all down one at a time. And it is kind of a tedious process, but it's one that you can get into the rhythm of doing and do it really well. And then also make sure you always do it without making mistakes. So the question again that we originally asked was, which percentage of offspring can be predicted to have freckles but no dimples? So I'm going to ask that you drag the orange stars to the offspring that have freckles, okay, have at least one capital F, but no dimples, which means they're going to be lowercase d, lowercase d. You may or may not use all of the available stars. So out of the ones that we're looking at, we have 4 out of 16 that are actually going to be like this. So you should have marked these 4. Capital F's present in all of these, good. But they also have to have lowercase d's, no capital D's. And so this row is the only row that has that. So the percentage is going to be 4 out of 16, or 0.25 times 100%, which would be 25% chance. So every time this couple has a kid, they have a 25% chance of having the kid that has freckles, but does not have dimples at the same time. So let's go through a practice problem together using the rake method. Here's our question. Let me move it out of the way. The inheritance pattern for an autosomal dominant trait is shown in the pedigree. Shaded symbols represent individuals that express the dominant trait. Okay, so based on this pedigree, what are the most likely genotypes of individual I1 and I2? Well, let's highlight the important parts that we got out of here. So we know that it's a dominant trait and that the shaded ones are going to be the dominant ones. So the specific things we're looking for are what is going to be I1 and I2's genotypes. Well, we're going to make a prediction now. We do realize that I1 is shaded in. So the man here has the dominant trait, but the woman here does not. So what is the question actually asking? Using this pedigree, what is dad's genotype and what is mom's genotype? Well, using this pedigree, we're going to have to figure out what knowledge we have on this pedigree. And what we can see is that mom's circle is not filled in, which means she does not have a dominant trait, which means she has to be recessive, right? Dominant means that it'll show even if there's just one single copy of that allele. So it means she has to have both recessive alleles. But the dad's square is filled in, so he has a dominant trait, but we're not really sure exactly if he's capital A, or capital A, lowercase a yet. So now let's eliminate the nonsense answers. Right? We want to look at the moms, right? So the mom is I2. So I2, if the mom is little a, her answer choice can't be F, G, or H. Let me move that out of the way when we actually do it, right? The mom has to be lowercase a. So therefore, our answer choice is going to be H. And that's an easy way to solve this question without worrying too much about what the dad's genotype is. So now you're going to work on your own. You have some questions to go through using the rake method. Remember to do all the different parts of it. You got this one, this one, this one, and this one to work through. As you finish your pair deck, make sure again you filled out all of the rake method stuff for your completion points. And then you're going to go ahead and do your exit ticket.